Is it on now, the microphone? Thank you. So good afternoon, everybody, and welcome to the session on tissue engineering for drug development. We have nice topics this afternoon. We will hear talks about bioprinting, about organs on chip, about multi-organs on chip, about stem cell-based micro tissue engineering, and also about smart and living implant materials. My name is Markus Rimann, and I will guide you through the session this afternoon. I will also give the first presentation. I'm working for more than 5.5 years at the Zurich University for Applied Sciences, and I'm working in the group 3D Tissues and Biofabrication. So we are interested in developing 3D cell culture systems with different technologies and also with bioprinting. So I will talk about bioprinting today. So we have a bioprinter in our labs that is encaged into a sterile printing environment. We have cell-compatible print heads that are based on inkjet and extrusion print heads. We can print into standard cell culture devices, as you can see here. So this is the bioprinter encaged in this fume hood, in this laminar flow, and we can print up to in 24 well plates. We also develop printable materials, so-called bioinks in this context, and they are mainly polymerized through UV polymerization. As you can see here, this is a UV LED integrated. It has a wavelength of 365 nanometers, which is not really cytotoxic to the cells. We also integrated into the bioprinter a cell mixing system to avoid cell sedimentation during the printing process. One of our projects we have is to develop a bioprinted muscle tendon tissue. So our industry partner from Novartis was interested in developing a novel in vitro system to assess drug effects on muscle tendon tissues. The problem is that the standard experiment they use at the moment is an ex vivo experiment where they have to isolate muscle tissues from mouse or rats. They have to put it into a small device, culture it under certain conditions, and then measure the contractile forces, which is really cumbersome, not really reproducible, and very time-consuming. Our idea is to come up with a solution where we have 2 posts in a well, as you can see here, and in between we would like to form a muscle fiber. In order to standardize it a little bit better than the ex vivo system, we would like to use bioprinting. So this is a schematic drawing of such a layer-by-layer system. Usually we print 1 layer of bioink without cells. We print an additional layer of cells, here shown in red in this rectangular shape. And then we alternate these 2 layers to pile them up and form a multi-layer tissue that is afterwards matured in the incubator. What we also develop in the frame of this project is a multi-well device where we can not only produce the tissue, we can print the tissue in there, but also to have a functional readout in the same well plate. So this is such a 24 well plate with 2 post holder inserts in there in blue. We have 2 posts in there where we would like to produce the muscle fiber in between. We have 2 concepts, 1 post with more a rectangular shape and the other one with more a round shape. From the top view, you can see that you have a fenestration in here that allows us to monitor tissue generation with an inverted microscope from below. We have an extended needle to print directly into the well plate around the post and in between the posts. And in a follow-up project, we would also like to integrate electrodes, here shown in red, into the lid of the well plate to electrically stimulate the muscle tissue and also to measure afterwards the deflection of the posts when the muscle is contracting. So these are the cells we are using. We use a primary human Myoblast here. We print them usually in this dumbbell shape here, so you have 2 holes in here for the posts. We printed them, we differentiated them, and then we did a myosin heavy chain staining, which is typical for muscle differentiation. And as you can see in green here, we have this positive staining for MHC, and you also see myofiber formation. It's not completely aligned as you expected in a native muscle, but it goes in the right direction. The same we did for primary red tenocytes in this case. We printed them, we differentiated them in the same pattern, and we could see afterwards that we have this typical collagen 1 dotted pattern around the nucleus of differentiated tendon tissue. We also saw that we have this typical striation for the muscle tissue of skeletal muscle, and we also see that we have myotube formation, so the fusion of cells into multicellular cell bodies. Of course, it's very important that when we are talking about muscle, it needs to be able to contract. So we printed these ones, we differentiated them, and then we applied an electrical stimulus. And as you can see, it looks a little bit like breathing, so it's really contracting the muscle. So this would be between the post, and the post would be more or less here on one side. So we could show that we have a biological functional muscle after the printing and after the differentiation process. We also analyzed some preliminary gene expression profiles where we could see an upregulation of myotube markers for the muscle tissue, but also an upregulation of tendon markers. I don't want to go too much into detail of that. The final goal of the project would then be to co-print the two cell types. So the tenocytes around the posts, here shown in green pre-labeled, and the myoblast in between the posts, that you can really have a tendon that is attached to the posts, representing the bone, and in between you have muscle fibers. As you can see, it's not completely perfect, the printing pattern, so we have a lot of green cells in between as well, but we are still optimizing the printing to be able to really have a nice separation and to have at the end the right connection between the two tissues, the myotendinous junction. Of course, what's also important is that we can not only co-print the cells, but also co-differentiate the cells. These are precursor cells, they need to differentiate, and media composition is always an issue when it comes to two cell types that need to differentiate into two different tissues. We could develop a media that allows the differentiation of printed myoblast into muscle tissue and also the tenocytes into tendon tissue. This is again MHC staining, myosin heavy chain in green, and this here is collagen 3 staining, so we have fiber formation of functional tendon tissue. So we could differentiate these cells into the corresponding tissues after the printing process. With this, I would like to summarize what I just told you. So I hope I could show you that bioprinting is really a nice technology. It's an enabling technology that allows you to create more or less complex tissues that are, in my opinion, at the moment more suitable for drug development and substance testing than for regenerative medicine. But as soon as the technology develops, I would see lots of application in regenerative medicine. We also developed a specialized well plate, not only for tissue production, but also for the readout, for analysis afterwards in the same well plate. We are also constantly developing novel bioinks that are optimized for printing purposes in terms of rheology, but also optimized for specific cell types, that we can really have the right cytocompatibility for the tissue we would like to generate. I showed you a proof of concept of printed muscle tendon tissues. Before, we also did the full thickness skin model, which I did not show here. And I would also like to show you or to introduce you to this competence center, TED. So this TED has also was involved in the putting together this session of the speakers. So it's called TED, Tissue Engineering for Drug Development and Substance Testing. It promotes the 3D cell culture from the basic research through applied research, clinical research, spin-off startups are involved, but also the big pharma industry, medtech, and cosmetic industry. So we have lots of activities, scientific workshops we organized, or symposia. The next one, which we co-organize, is called the Biointerfaces International end of August in Zurich this year. It's organized through an NTN, a National Thematic Network, led by Professor Graf Hausner, and we have lots of partners from Switzerland, but also internationally from Europe. Lots of industries involved, as you can see in red, more than 50%, and we have a membership fee of 500 francs per year, so it's not too much, but it really helps to bring together the right people throughout the whole value chain of 3D cell culture. With this, I would like to close my talk and thanks the ZAW team and our partners, Retinue, for the bioprinting expertise, Wädenswil for the well plate development, Novartis for their tissue engineering competence on muscle and tendon tissue, and the CTI for funding. And I would like to thank you for your attention. Thank you. So the thing is, we would like to postpone all the questions to the end of the 5 talks. So if there is no immediate question, I would rather like to continue now. Is that okay? Good.